Okay, so last week when we were talking about the ways that cells transport material, we said there were the big three, okay? The big three methods of cellular transport. Osmosis, which we said transports only water, okay? And it moves across the membrane, and that's in order to balance salt concentrations, solutes, okay? And it's passive. Okay, so this one involves only water, and it involves going across the membrane. Diffusion moves anything within the cell, not across the membrane. Okay, it's passive. It only moves from high to low, and it's slow, and it's not targeted. Okay, which means it doesn't. There's no guarantee it actually ends up where it's supposed to. It does most of the time. Okay. Now, the other problem with diffusion, what did we say? That diffusion works well within the cell, but it only works well over small areas, short distances. That's what we want to look at a bit today. Okay, and then active transport, okay, it uses energy, it moves against the gradient, and it's also across the membrane. So what we're looking at today is the cell's ability, or this activity is going to have to do with the cell's activity to transport material within the cell. So which transport method are we looking at? Diffusion. Okay, we're only looking at diffusion here because we're talking about the cell's ability to transport material from place to place within the cell. Right? And the only way that that can be done is by diffusion. There's no intracellular transport other than that. All right, questions so far? Okay, now I don't think you have this anywhere, but it's just something we're going to talk about here. Okay, cells absorb nutrients and they excrete wastes through the membrane. Okay, and that's how stuff gets in and out. We talked about that. That's endocytosis and exocytosis. Okay, but they have to transport through their cytoplasm, okay, or their internal volume. Okay, and we've said that that's accomplished by, how do they transport once it's inside? Diffusion. Okay, so as a cell size increases, the volume increases faster than the surface area. Okay, so the membrane represents the surface area of the cell. We'll say it's a sphere. Okay, and as the cell gets bigger, the surface area increases, but the volume increases even faster. Okay, in fact, exponentially faster. If you're going to calculate, let's say, the area of a cube, how would you do it? No, that'll get me the volume. Length times width times height will give me the volume, but we'll get to that. Length times width times height. How do I get the surface area of a cube? Length times width times the number of sides, in this case, 6. Okay. All right. So really, when I'm calculating surface area, or I'm looking at surface area is two-dimensional. Okay, everyone follow me there? It's two-dimensional. It's flat. Okay, if I just undo the cube and lay it out, I get a square. All right, but when I'm looking at volume, it's three-dimensional. Okay, this 6, that's never going to increase as long as I'm dealing with a cube. Agreed? Okay, so I'm just going to get rid of the 6 for now because it's never going to change. It's always going to be the same number. If I make my cube bigger, this number gets bigger, and so does this number. Length and width both get bigger, right? And I multiply them together. But as my cube gets bigger and I'm calculating its volume, all 3 of these numbers get bigger. So which one gets bigger faster? Volume. Okay? It gets bigger faster because I'm always multiplying by another number that's also increasing. Okay? So as a cell grows, its internal volume becomes very big, very fast. And that is going to lead to the cell having some difficulty, because now the distance over which diffusion has to act has become much, much greater. And we've said diffusion is only good over small distances. All right, so here's what we're looking at. Let's say we've got these two cells. Okay, they have the same number of organelles, which I've represented with x's and squares. And the nucleus is the circle, because I can't draw. Okay, and I'm not going to embarrass myself by trying to go any better than that. In a small cell, you can see that the organelles are not very far apart. So material doesn't have to transport very far by diffusion. But as the cell gets larger, it's not that it's not going to be making a whole lot more organelles. It's going to have pretty much the same number of organelles as the small cell did, okay, or as it did when it was small. But now the distance between them all has increased a great deal, right? And so it's going to take much longer for the stuff to get from one thing to the other. It's kind of like going through Calgary, okay? If you came here, let's say like in 2000, Calgary was much smaller, okay? Deerfoot extension wasn't even finished. In fact, it wasn't even being thought of. Deerfoot only ran to about Southland, okay, at that time. There was really one way to get from the north end of Calgary to the south end of Calgary. You took Deerfoot, and then you went to Southland, and then you went like McLeod out through the south end. It was terrible, okay? It took quite a while to get through. And there was one way east-west, and that was Trans-Canada, okay? 16, or Highway 1 there. Everyone with me there? Okay. Because Calgary was smaller, you know, it wasn't too bad. But as Calgary increased in size and before they got all those new roads built, it took a lot longer to get through Calgary. Okay. When I was your guys' age, it took maybe about a half an hour in bad traffic to get through Calgary from one end to the other. Okay. Now, okay, Calgary in bad traffic, if you're going the Deerfoot route, sometimes that can be an hour and a half. Okay. Because Calgary is bigger, right? And there's only one way. If we imagine Stoney didn't exist, there's only one way to get through it. Okay, so as something gets bigger, it's just going to naturally take longer to get through. All right, is that making sense? Especially if you only have one way to do it. And that's kind of the problem a cell has. It only has one way to move stuff from one place to the other, and that's by diffusion. Okay, and diffusion, although free, has a few drawbacks. All right. So the problem we're going to be investigating here, how does the surface area relate to volumes in cells of different sizes? Does dividing a large cell into smaller cells affect the surface area to volume ratio? That's what we're going to try and find out kind of mathematically here. All right, so you need a hypothesis that talks about the relationship between surface area and volume in cells and how that can affect the functioning. So the if part of your hypothesis, okay, should probably mention something about how a cell transports material. Okay. The and part can mention cells of different sizes, something about what we're going to do here. We're using imaginary cells, these three. Okay. Okay. And the then part should talk about what will happen as a cell gets larger. Okay, do I already have the chart built on your guys' template? Yeah? Okay. All right, we're going to fill out that chart together. So basically, we're going to do the math together here. So you're getting like 5 free marks on this one, because it's actually out of 5 to do the chart. Okay? We're comparing these 3 cells here. Okay? We've got one that's called micro, and that's a really small cell. Its dimensions are 1 by 1 by 1. Okay? We're looking at macro, which is a very big cell, whose dimensions are 10 by 10 by 10. Okay? And then we're looking at multimicro, which is not a single cell. It is many cells. It is, in fact, macro, but divided up into micros. And not 100 of them, but 1,000 of them, because its dimensions are the same, 10 by 10 by 10. So ignore these hundreds. I thought I had erased those. They were in pencil, but for some reason the photocopier's scanner was exceptional that day, and they still showed up. Okay. All right, so it's 10 by 10 by 10, but it's divided up into little macros. Okay, the whole box is, or sorry, micros. It's filled up with these little tiny micros, 1,000 of them. All right, remember, these are 3-dimensional objects. Okay. So let's look at figuring out the surface area of micro. How would I figure out the surface area of micro? 1 times 1 times 6. Okay, we figure out the surface area of one side and multiply it by the number of sides. So we are going to say that the surface area of micro is 6 micrometers. 1 times 1 times 6. Okay. And what's its volume going to be? Remember, it's 1 by 1 by 1. So its volume is going to be 1. Okay, now we also have to say that this is 6 micrometers square, and this is going to be 1 micrometer cube. All right. Does that seem like a positive situation? More membrane than there is cytoplasm. Would that be good from a transport point of view? Absolutely. Okay, if you've got tons of surface area compared to your volume, you can acquire tons and tons of stuff through your very large surface area, and you don't have to transport it very far because you have a tiny volume. Okay, that's an ideal situation. The surface area to volume ratio is 6 to 1. Basically, any time the surface area is larger than the volume, you're in pretty good shape, especially if it's quite a bit larger. And in this case, it is 6 times more. All right, macro. Macro is 10 by 10 by 10. Okay, so what's its surface area? 10 times 10 times 6. So 10 times 10 is 100, times 6 is 600 micrometers square. What's its volume? Thousand. 10 times 10 is 100, times 10 is 1,000. 1,000 cubic micrometers. What's our ratio? 600 to 1,000, or let's reduce that down. It's 0.6 to 1. Does that seem like it would be very good? We have comparatively little surface area to large volume. Okay, quite a bit more volume. All right, multi-micro. This is the tricky one. What's the surface area of multi-micro? It has the same dimensions as macro. Right, micro times, how many are there in there? 1,000. Okay, remember, it's a three-dimensional object, and it's made up of smaller three-dimensional objects. Each and every micro that multi-micro is made up of has 6 sides. Okay, now a lot of people say at this point that Codair, a whole bunch of them are inside. They don't really count towards the surface area, but they do. Because in a real organism, your cells are stacked all together in a three-dimensional space too. Okay, but they all still have access to interstitial fluid. That's the fluid that surrounds all cells. So the cube is a little bit artificial in that it looks like it's sealed tight and the cubes that are inside wouldn't be able to access anything, but they can. All right, so if every micro has a surface area of 6 and there's 1,000 of them, multi-micro's surface area is 6,000 micrometers. Okay, square. What's its volume going to be? $1,000. That doesn't change. It's 10 by 10 by 10. Okay. So 1,000 cubic micrometers. And so then its ratio goes to 6 to 1. Now, does that make sense? I mean, if I'm a multicellular organism, I'm made up of a whole bunch of cells. My overall surface area to volume ratio should be the same as each individual cell. Okay? Is that why it's better to be multicellular than it is to be single-celled? Yeah, that's the only way you can effectively get big, okay, and not have this problem. If you're a single cell and you get to this size, you're going to struggle. But as a multicellular organism, size is no longer a problem because you're made up of these small things. Everybody follow where we're going there? Okay. So you've got some analysis questions to do. Okay, there are 7 of them. Okay, don't miss the last ones. And then you got conclusion. There's no sources of error. Okay, there's also no design, if you notice that. So this is like a short activity. That's why I said realistically we should be able to get it done before class is over. All right, I'm not saying it's due at the end of class, it's going to be due on Wednesday, okay? But I'm assuming that we can easily get this done in class, because really all you got to do is make a hypothesis and answer some questions, okay? All right, so I'm going to leave those questions to you. Oh, sorry, one thing about the analysis questions here. The wording is weird because I took this from a book and modified it a little bit. Okay, when the cube dimensions increase from, it says centimeter to centimeter, we're using micrometers here. Okay, so essentially they're saying when the cube increases from micro to macro, the increase is a factor of 10. Okay, what's the factor of the increase of surface area and volume? So basically they're saying how many times bigger is the surface area of macro than micro? How many times bigger is the volume of macro and micro? They're just asking you to compare, okay? And number 2 is the same thing. They're asking you compare macro and multi-micro, same idea. And then for number 3, that one you guys can figure out. Now, number 4 is asking about these diagrams right here. Okay, you have a liver cell, which is basically a sphere. Okay, it's almost perfectly round. Okay, you've got skin cells here, which are kind of more like this, okay? They've got one side that's noticeably flatter than the others. And then you've got the nerve cell here, and the nerve cell is quite a bit more like this, okay? In fact, there are nerve cells that run from the base of your brain to the small of your back. One cell, okay? You actually have cells in your body that can be up to a meter long, but they're extraordinarily skinny. Okay, so what we're looking at here is, does shape of a cell affect its surface area to volume ratio? Okay, can I make a cell be really big in one dimension but still retain a good surface area to volume ratio? Okay, that's what 4 and 5 are asking you to do. Okay, you will have to do a little bit of measuring, except that I didn't give you a paper copy. So I'll let you get started, and I'm going to put the dimensions for these up here on the board so you know what they are. Okay? No, you do not. There's no design part, so just don't bother with the problem. Okay? All right, you guys can get going on that, and I'll get the measurements done here for these squares here. Okay, so the dimensions are up here, okay, for those 3 cubes. So cube A is 1 centimeter by 1 centimeter by 1 centimeter. Okay, cube B is 1 centimeter tall, okay, 0.5 centimeters wide, and 2 centimeters deep. Okay, the cube C here, or box C, it's not really a cube, I guess, okay, is 1 centimeter tall by 0.1 centimeters wide by 10.7 centimeters deep. Okay, so you can use those dimensions in your calculations. All right, saves you the measurement. When you're calculating the surface area of A, you can obviously go 1 times 1 times 6. Can you do that for B and C? No, because the sides aren't the same. So you got to calculate the surface area of the front side. The back that you can't see is the same, so multiply it by 2. Calculate the surface area of this side, multiply it by 2. Surface area of the top, multiply it by 2, and add those numbers together. Okay, that's how you get the correct surface area for shapes that are irregular. All right. I didn't give you the answer, I gave you the dimensions. I gave you the dimensions so you could calculate the surface area and volume of each one of those. Yeah?